welcome you. I welcome everyone to the second webinar. As you know, this is the EIT Food Grow initiative, where Food for Sustainability is a proud partner of this great initiative. And this series is part of our ongoing efforts to promote sustainable food systems through education and innovation, as you know. And in the previous session, if you remember, for those that were here, we laid the groundwork by exploring the fundamental concepts of biostimulants and their mechanisms of actions. If you remember, we also touched upon seed microbiota and its potential applications across related crop families. And we ended our session last week looking at the crucial role of formulation in biostimulants effectiveness. So today we're going to build on these concepts, and I'm trying to move forward my slides. So we'll build on these topics and delve deeper into practical applications and innovations. And we are very privileged to host three experts who will expand on these topics. And I hope they raise your curiosity and that you have loads of questions to our experts, which I very briefly introduce to you. So first we have Sharon Doty from University of Washington, and will be presenting Help from Within, Harnessing the Power of Bacterial Endophytes for Nutrient Acquisition and Resilience to Abiotic Stresses. So Sharon, she will share her work and showcasing how internal plant microbiomes can enhance crop resilience and nutrition. Thanks, Sharon, for being here with us. Then we have Jacob Malone from the Joint Hines Center. And apologies, Jacob will revisit and expand on the previous discussions we had on seed microbiota with his talk on seed microbiota as a new promising source for next-gen biostimulants. And Jacob's research opens the new frontiers in leveraging seed-associated microbes for a more sustainable agriculture. And we will end today's sessions with Rosaria Campilongo. She is the CEO of PfBIO, and she will share her journey from research to market, advancing biological solutions to promote sustainable agriculture. And Rosaria's presentation will bridge the gap between academic innovations and real-world application in the biostimulants industry. I am sure that these presentations will offer a comprehensive view on how microbiome-based solutions are shaping the future of agriculture, building on the foundations we have established in our previous webinar. So again, thanks for being here, and a great thank to all our presenters. And I will just take a moment to introduce you, for those that don't know, our Soil and Function Laboratory. As you know, this is a state-of-the-art facility dedicated to advancing our understanding of soil health and its critical role in sustainable agriculture. And as you know, our lab combines cutting-edge technology with expertise in soil microbiology, biochemistry, and ecology to provide a comprehensive analysis of soil microbiomes, microbes, and their functions. And through our work, what we aim is to bridge the gap between scientific research and practical applications, offering, of course, valuable insights to farmers, agronomists, and researchers alike. As always, we are fully available at Huda Lab at foodforsustainability.org, and I'm sure that Soraya will post here our email address for you if you want to know a little bit more. I'm also excited to announce that the registration for our summer school on soil health are now open. Let me just share with you that this intensive program offers a unique opportunity to deep dive into the world of soil microbes, understand also their impact on plant health and innovative approaches to sustainable agriculture. That's really what we need and what we are here for. So participants in this program will not only learn from leading experts in the field, but they will also have the chance to develop their own ideas and projects under expert guidance. This program is aimed at whether you are a student, a professional in the agriculture sector, or simply passionate about soil health. This summer school promises you an enriching experience. So the registrations are now open. Please join us to be at the forefront of soil science and sustainable farming practice. And again, all together, let's try and shape the future of agriculture. And I will ask Soraya also to share with you the link here in the chat. So after this very short introduction, I think it's now time to do our warm-up as if we are going to enter into kind of a tournament. I believe that Soraya has already shared our Slidos, Soraya. So I will invite you all to jump on Slido. Okay. You know, Slido is fairly easy to join. You can scan the QR code with your mobile or use a browser, your browser in a tab, slido.com. You'll have a window with an hashtag. Just type in F for S Academy and we can see what the team has prepared for us. Okay, let's see. Let's start with our knowledge, okay? So I think by now we are all able to answer this question. So first of all, I invite everyone to jump on Slido, but the question is what percentage of all plant species interact with mycorrhizal fungi? Okay, first we need you on Slido, then we need you to start answering the question. So plant species that interact with mycorrhizal fungi. Half of the plant species, 3/4 of the plant species, 80% of all plant species, or are we talking about more than 90% of all plant species interact with the mycorrhizal fungi? This is a type of fungi that I'm sure is going to be addressed in today's session. They are always coming up. Surely they are very important. Okay, so we have a stabilized answer, and you are absolutely right. Over 90% of plant species have this secret ally, the mycorrhizal fungi. And these tiny fungi form symbiotic relationships with plants, helping plants to absorb water and nutrients from the soil. As we all know, this is a partnership that is crucial for plant survival and has been a foundation of the success of land for millions of years. Thank you very much for taking part. Now we're going to go with some definitions. So the question is, what is the word for a plant that works as a team with its microbiome to survive and adapt to challenges? And we have here phytobiome, symbioplants, bioflora, holobionts. Okay, so what is the word that relates to when a plant is working with its microbiome? Okay, let's vote. I will share with you some definitions because I think this is also an opportunity for us to learn a little bit more. So phytobiome refers to the entire ecosystem associated with a plant, including the plant itself, its environment, and all the microorganisms living in, on, and around it. So it's like a plant's whole world and all its inhabitants. So this is the phytobiome. Symbioplant is a term that is not used widely in the scientific community. It combines symbiosis, living together, and plant, suggesting a plant that lives in close associations with other organisms. Bioflora. This isn't bioflora refers to the collection of plant life in a particular region or habitat. It's more about the plants themselves rather than the relationships with the microbes. And the holobiont is the combination of a host organism, for example, a plant, and its associated microorganisms, its microbiome. It views the plant and its microbial partners as a single ecological unit that works together to survive and adapt. So, after this, we know that the correct answer is holobionts. Indeed, this concept emphasizes that plants and animals, including humans, are not isolated organisms, but they are instead a complex system that includes that microbial partners. And this view is changing how we understand ecology, evolution, and health. So when we look at plants and working with its microbiome, we are referring to the holobionts, okay? I think we learned something already. Thank you so much for these questions. I think we have a final one. So do you have an idea what region or country is leading the current biostimulant markets? The country or region leading biostimulants market? Europe, the US, Japan and China, or India? Okay, give you a couple of seconds to... Okay, this is interesting to see the perceptions. Okay, Japan and China leading in terms of biostimulants. Okay, China is definitely at the forefront of a lot of our industries. Come on, okay, we are about to... Okay, so we have a clear winner, but the region that has adopted the most research and use of biostimulants is Europe, particularly driven by countries like France, Italy, and Spain. Europe represents around 50% of the global biostimulants market and is expected to continue growing due to the strong focus of Europe on sustainable agriculture and its stringent environment regulations. So Europe at the forefront leading the biostimulants markets. And this comes to the end of our warm-up. It's time for us to begin the exploration of this exciting topic that we have here. So I will call to the floor Sharon Doty while I properly present Sharon. Sharon has received her bachelor degree in genetics with an emphasis in botany from University of California, Davis. Sharon has earned her PhD from the microbiology department at the University of Washington, studying agrobacterium plant signal perceptions and responses. Her postdoctoral research was in plant biochemistry, developing plants with improved phytoremediation capabilities. Her research focused on the importance of the plant's microbiome for plant growth, nutrient acquisition, and tolerance to both abiotic and biotic stress. Shannon, the floor is now yours, and thank you so much for being here.
Thank you for the opportunity. Let me share my screen. Okay, I assume you can see that. So I'll be talking about help from within, harnessing the power of bacterial endophytes for nutrient acquisition and resilience to abiotic stresses. Okay, so first, just description of what endophytes are. These are the microorganisms that live inside of plants. So endo is within, phytes is plants. So they can be within the leaves, the branches, and the roots. They have direct interactions with the plant tissue. They can be selected for by the host plant, and they can be very stable. So these are distinctly different from the rhizosphere, which are bacteria that are in the soil that it's near the roots. So endophytes are inside of plants. So it's recently been understood that the human microbiome is very important for our health. And I believe that the plant microbiome is even more important for plants because plants can't move. So, especially trees that are in the same spot for decades, they need a nutrient source for all of that time. And if there's an environmental challenge in that spot, they can't get up and leave. And so I think that they depend quite a lot on endophytes to help them to deal with these challenges. My lab works on all of these different ways that endophytes help to promote plant health and growth. They can provide resistance to pathogens by producing antimicrobial compounds. They can even detoxify environmental pollutants. But today I'll talk about nutrient acquisition and a little bit about abiotic stress tolerance, like drought and temperature stress. So first of all, about nutrient acquisition. So plants are very dependent on their ability to access nitrogen in particular, also phosphorus and potassium, but primarily nitrogen. And so it's common to provide that resource through chemical fertilization. So we can produce nitrogen fertilizer, but it's a very costly, high energy process. So that it's from fossil fuels and high temperature and high pressure. Then we can make ammonia. Um, but that has not only financial costs, but it also has environmental costs because this can leach into the waterways and disrupt aquatic ecosystems. And it also the excess nitrogen that's in soils can be converted to nitrous oxide, which is a potent greenhouse gas. So instead, we can use biological nitrogen fixation. So this is a natural process. The air that we breathe is about 80% dinitrogen gas, but it's inert. It's a triple bond, and it's difficult to break. And but there are some bacteria that can fix that into usable forms, and they do that through the action of the nitrogenase enzyme. So they can reduce that dinitrogen gas to ammonia. So that's the form that plants can take up and use. But this process is energetically quite expensive for cells. And so it's often that these bacteria will associate with plants because plants are doing photosynthesis and they're providing sugars. So they can release that in exchange for the nitrogen that's being fixed by the bacteria. So this has been a really well-known symbiosis with legumes and rhizobia that fix nitrogen. So legumes are things like peas and beans. If you look at the roots, you'll see these structures on the roots, and that's where these bacteria are fixing nitrogen. It's a very specific interaction between a particular species of legume and a particular species of nitrogen-fixing bacteria in the system. Another common example is with actinorhizal plants and Frankia. Frankia is another nitrogen-fixing bacteria, also forms these root nodules. They look kind of different and they're not related, but they're the same idea where the bacteria is fixing nitrogen in these little houses. And so for 100 years or so, people have been thinking that that plants are only symbiotic with nitrogen fixing bacteria if they have these root nodules. But that is not true. It was in the 1990s that it was shown by Dr. Johanna Dobreiner of Brazil that sugarcane in Brazil has bacterial endophytes that can fix nitrogen, and they are not housed in root nodules. They're within the plant tissue. So they can be within the leaves and within the roots. So these bacteria can fix nitrogen to high levels. So that was just the 1990s, so fairly recently. My research began in, it was about the year 2000 or so, when I discovered that there is rhizobium inside of poplar trees. So poplar is, Populus is the genus. I work with one called poplar, Populus trichocarpa, or black cottonwood. And then willow, and that's Salix is the genus. These are early successional pioneer plant species, and they're able to colonize just really bare substrates. So they're pioneers. They colonize this new land that's produced upon flooding of a river. Our riparian zones here in Washington State are not like, it's not like the Amazon River or the Nile River, where it's bringing in rich sediment. In this case, it's just rocks and sand. Our rivers are from high alpine snowmelt. And poplar and willow are not nodulating species. So they don't have that type of symbiosis, and yet here they are in this just rocks and sand. So what we discovered was that there's nitrogen fixation going on inside these wild poplar trees. So we took pieces of the poplar and enclosed them in flasks, exposed them to a stable isotope version of dinitrogen gas, so we could measure incorporation. And so using that assay, as well as other methods, we demonstrated that there is indeed nitrogen fixation going on inside of these poplar trees. We've also done the microbiome analysis and showed that there are a lot of different nitrogen fixing bacterial species inside these wild plants. So that was published almost 10 years ago now, but it's taken a long time to get people to recognize that. So recently we got a grant from the United States Department of Energy to study this, study the mechanisms and look at more detail. So we found that the nitrogen fixation is occurring, but it's in a subset of the cells. So about 10% of the population of a particular strain of bacteria is actually fixing nitrogen, and the rest of them have a supporting role. Recently, we discovered that these bacteria are fixing nitrogen within these like capsules. We call them nitrosomes inside a plant cell. So this whole thing here is a plant cell, and then these are these little capsules, and the green is showing nitrogen fixing bacteria. So it could be that this is like towards the evolution of a nitrogen fixing organelle inside of these plant cells. So the neat thing about endophytes are that they can be cultured. So unlike soil bacteria, where only about 1% are culturable, with endophytes, it's about 70% are culturable. And so we can grow them up, we can do these studies, and we can add them to other plants. So we took poplar and willow from took the microbes from poplar and willow in this wild environment and added them to cultivated poplar, and then found increased greenness, more chlorophyll, higher nitrogen, and a doubling of root mass. Then because poplar and willow are not legumes, and yet there's these nitrogen-fixing bacteria in them, we looked to see if they could colonize and fix nitrogen for other plants. So we found that they do colonize rice, and they have increased biomass if they're given these bacteria grown under nutrient-limiting conditions. And same even for forest trees. This is Douglas fir, Pseudotsuga menziesii is the scientific name. And under nutrient-limited conditions, the inoculated trees grow better. You can see increased root mass as well. So that was about nitrogen, but plants also need phosphorus. That's another macronutrient. So we also got bacteria from these wild plants and showed that they can dissolve the phosphate make it more available to plants to take up. So you can see that with this clearing of the phosphate mineral on these plates. So they can make this essential nutrient more bioavailable. I've licensed these bacteria out to a company called Intrinsics Bio that now works with Syngenta. They did third-party validated greenhouse and field trials using the endophytes from my lab on a wide variety of crop plants and showed increased growth with reduced fertilizer requirements. These endophytes have a very broad host range, so it's quite remarkable that they can be taken from these what are called eudicots, type of angiosperm, and they even work on gymnosperms, even though those diverged millions of years ago. So I think that this is showing that it's a natural part of the plant microbiome, and it's something that we've just lost the way that we do common agriculture, because these the plants are all recognizing these as helpers, parts of the natural microbiome. So I see I just have a few more minutes left, but I'll just briefly touch on stress tolerance conferred by these bacteria. So in these experiments, we did a consortium or a mix of about 10 of these strains that we were studying that can fix nitrogen and solubilize phosphate and also produce plant hormones. We found that they have this other remarkable impact, that they can help with drought tolerance. So this particular forest tree, Thuja plicata, is very sensitive to drought. This graph is showing mortality. So under drought conditions, 10 out of 10 of the plants died. But if they were given these endophytes from poplar and willow, the wild ones, then only one died. So it's the difference of life versus death. Same sort of thing with hybrid poplar. They were able to survive even a whole month without water. Our current research is looking at the mechanisms involved, and I don't have time to go over that, but into that
we still
later.
have a couple of minutes, Sharon. We still have a
Okay.
couple of minutes. You can you can detail if you want.
Okay, great.
Yeah.
Thank you. Okay. Yeah, so we've been looking at how they do that, and it turns out that there's reduced stomatal conductance. So if you're familiar on plant leaves, they have stomata that are like the mouths for gas exchange. And so the plant needs to open those up in order to get carbon dioxide to do photosynthesis, but at the same time, they're losing water vapor. And we found that these bacteria are influencing those stomata. So they're smaller and they're able to close rapidly. And so overall, they're increasing the water use efficiency. We found that the bacteria are helping the plant's cells to be larger. So that ends up with increased overall leaf size and increased photosynthesis. In addition to drought stress, they also are helping with transplant stress tolerance. So we found that it's much more survival if they've been given those endophytes. So it's really neat. We've also recently moved on to heat tolerance because of climate change. And so we isolated a different set of endophytes. These are from poplar trees that are growing in these really arid environments, along with sagebrush and other indicator plants of that type of environment. We isolated endophytes and then tested them in the system where the plant roots are in water. So water is not limiting, but hot air is being blown on them. And so through those screenings, we identified 14 bacterial strains that help with heat tolerance. This one in particular is a Pseudomonas putida. And you can see the difference between uninoculated control, where many of them died, versus the ones that were inoculated are still alive. So we're looking at the mechanisms of that. So to wrap up, there's potential here for climate change mitigation and plant adaptation. Trees can take up carbon dioxide from the atmosphere that's causing the greenhouse effect and climate change. But photosynthesis then becomes limited by nutrients and water. So these bacteria can provide nitrogen and phosphorus, so they can take care of the nutrient acquisition. They can increase water use efficiency and drought tolerance, so then they can help the plants to continue doing photosynthesis. And they can also increase resilience to heat stress. So overall, I feel that the endophytes, especially from wild plants, are incredibly powerful. They can help plant growth and health under a wide range of stresses. So the work I shared with you has been over the last 20 years or so. So it's been under a lot of different funding. My current funding source is through the Department of Energy, and this is my current lab staff. So I'd like to help to thank them and to thank you for your attention.
Thank you so much, Sharon, and thanks so much also to be on time and to present us with what it seems to be just another option for farmers and food producers, to another tool to fight so much of the problems that are facing agriculture nowadays. It was a very interesting overview of bacterial endophytes. I will start by, using one of the expression that you had during your presentation. You mentioned that it took a long time for people to recognize. So the question is, at what stage are we in terms of farmers and consultants, understanding the value of endophytes? If on one hand, the rhizosphere starts to be kind of a more common word than the rhizobium and arbuscum mycorrhizal fungi, what about endophytes? At what stage
Yes.
are we in?
Yeah, I think that it's just now in the last year or so. That's how how recent it is. It seems to be catching on more now. So the major companies are now adopting this technology. So Syngenta, for example, um, and that was just December, and that now so they have these endophytes, and then my next set of strains. So yeah, that was a big deal that now it's finally out where it's going to be used. They've been doing a lot of field trials across Europe, especially a little bit in the United States. But as you said earlier, that Europe seems to be leading that market, and so yeah, it's been very satisfying after that first 15 years. It's been just such a
Yeah,
struggle to get people to
I,
even listen. It doesn't
I
have
can
root nodules, can't happen.
I can just imagine, how satisfactory it must be, after so many years of research and passion and dedication, to see products that are really at the service of solving a problem. And we will also have another example here today. It's very, very interesting. So there's already available commercial products. Do you know what is the main claim that these products do in terms of using bacterial endophytes? Because there are several of benefits that you mentioned. Do you know which one is the major claim that these companies use for farmers?
Yeah, I think it's the nutrient acquisition. And so, yeah, plants are healthier, they grow more, there's greater yield if they have these microbes. So we assume that's through the increased nutrients.
Okay, fantastic, fantastic. While our audience doesn't come to the fore and raise a question, I have another question. What are the potential benefits of using the endophytes to colonize rock and sands? You mentioned the Mississippi River, which from my generation we all know because there was Tom Sawyer on TV and all the adventures. But I wasn't aware of these characteristics. But I could straight away relate to some desertification areas, for example, that we are facing here in Portugal, in the south of Portugal, at high risk of the desertification and soil degradation. So do you think we can translate what you have found there in the banks of the Mississippi River to other areas that are facing the risk of desertification? And we can use the bacterial endophytes to stop this desertification?
Yeah, I want to point out, definitely not the Mississippi River here. That would be very, very different. But yeah, to be able to grow in just rocks and sand, it would certainly make sense that microbes from this type of environment would help with to stop desertification. The plants would be able to colonize that type of substrate. Yeah, makes a lot of sense.
Okay, so there is hope. Very, very good. So can you just finally just to highlight, you mentioned quite often poplar and willow as your experiments, but you also mentioned several other crops that you are using in your experiments. Can you just detail what crops have been the subject of these experiments?
Yeah, so I was testing rice and maize, turf grasses, tomatoes, bell peppers. But then when I licensed these bacteria out, then their focus was more on wheat and barley. And so we saw increased yields per hectare when they had these endophytes. So it is those
so it's a
major crops. Yeah.
Wide range of crops. Okay. One final question from Jacob. Are the other non-nitrogen fixing bacteria also relevant to biostimulation, or will this occur in axenic, example, hydroponic setups?
Yeah, so there are definitely other bacteria besides the nitrogen fixing ones. So we just recently published in ISME Communications that we found these synergists. So they're partners, bacterial partners to the nitrogen fixing bacteria, and they greatly amplify nitrogen fixation. So I think it definitely should not be just a single strain inoculum. Microbes work in communities, and you need to keep that community. And then also, we have different bacteria are better for the increasing the tolerance to heat. None of those were nitrogen fixing bacteria, so I think they have a different function mechanism.
Could I quickly follow up on that question? So it's fantastic work. So do you see differences if you look in the original community that the bugs came from? Do you get an enhanced response from your nitrogen fixers in the context of the original microbial community, or it's so long as they have a microbial community, will they provide the same benefits? Even if the rest of the community is non-native to...
Oh, right. Yeah. So these were all added to, in those like crop situations. There already is a native microbiome, so this is all being added to it. Yeah, so even the ones where it was poplar, hybrid poplar, they have a native microbiome, so the benefit was from getting these wild strains added to the ones that are acquired just from growing in potting mix, basically.
Fantastic.
Okay, great. Sharon, thank you so much. We need to move on. Thank you so much for your presentation. Please stay with us. We're now going to move to Jacob Malone. Jacob, I'll now call you to the floor. So Jacob is an expert in bacterial signaling and behavior during host interactions. Since 2011, his research group at John Hine Center in Norwich, UK, has used environmental and molecular microbiology, biochemistry, and systems biology to examine the complex processes of bacterial biocontrol and plant colonization and to understand the selective pressures that shape the root-associated soil microbiomes of crop plants. Malone has a strong track record of leveraging academic findings to enable translational plant and microbial research, with several industry-supported projects ongoing in his lab. In 2022, Malone has co-founded PfBIO, which is a spin-out company that seeks to exploit GIC research to enable effective crop disease biostimulation and biocontrol. Jacob, it's a pleasure to be here. We can see your presentation.
Right.
Yes, we can see your presentation in the presentation.
Fantastic. So I will just make a pointer here. Okay, thanks very much for inviting me. What I'm going to talk about today, the title is slightly different to the title that was presented at the beginning. What I'd like to do in the next 15 minutes or so is just talk about my the research that's ongoing in my lab, and how it relates to biostimulation, and ultimately how it relates to, or how we hope that we'll be able to commercialize this research in the future. So the title of my talk is Plant-Microbe Interactions for Sustainable Agriculture. So the work that we do in my lab is looking at interactions between plants and microbes in their environment. And the strong belief that we have in my research team is that context matters with plant microbe interactions. So understanding how plants work, understanding plants on their own is only part of the problem. Understanding how the microbiome works is only part of the story, and really understanding how all of these things work together is the key to getting effective plant growth. So we need to understand how roots will communicate with bacteria, how bacteria and other microbes will interact with roots, and how this will contribute, for example, to suppressing plant pathogens and enabling healthy plant growth. So we look at microbial communities, focusing on the pseudomonads, which are a major group of plant-associated growth-promoting bacteria. And we look both at the traits of these bacteria, so the different phenotypes that they show. I should say all of these are Pseudomonas fluorescens species taken from the same handful of soil. And you can see all of the diversity that's available within the soil microbial community. And we also look not just at their phenotypes, but also at their genotypes and the traits that are encoded in their genomes, and ultimately what these different systems do to enable colonization and plant growth promotion. So that's the research that goes on in my lab. But for most of the talk, I would like to focus more on the field in which we're working and how our research relates to the wider area of biostimulation. So if we're looking at plant roots, we have various different areas of the plant roots to consider. So we have the endosphere, of course, the internal spaces of the root, which Sharon has just explained her exciting research on. We also have the area which we focus on mostly, which is the rhizosphere and the rhizoplane, which are the areas of the plant that are directly affected and directly influenced by secretions coming from the plant roots. So all of these different areas have different communities of microorganisms associated with them. So the endosphere will have, for example, these arbuscular mycorrhizal fungi, as well as different bacteria which live in the interstitial spaces between plant roots, between the cells of plant roots. But then the rhizosphere itself is a tremendously complex environment containing a huge diversity and abundance of different microbes. And those microbes will have huge influences on the plant. The plant will select them, and then that selection is in order to promote the plant's growth. So when I say there's a huge diversity, that is in no way an exaggeration. This is a really old piece of research now. It's nearly 15 years old, and it's one of the the early studies looking at the diversity of taxa which you will find in soil surrounding plant roots. And we think this is now an undercount, but even back in 2011, they were able to identify 32,000 different taxa. So all of these are different species and strains of bacteria and fungi. Well, this is just the bacteria, I should say. You also have fungi and oomycetes and all sorts of other things. All of these different microbes, they're all interacting with the plant and they're all interacting with each other. And within each of these groups, you will have a tremendous diversity, both of form and traits. So all of these different shapes, for example, here with the actinobacteria, and also with the proteobacteria. As I said, for example, this is Pseudomonas fluorescens. This is one species within proteobacteria, which contains thousands of different species. So there's almost infinite variety and diversity in terms of bacterial traits, bacterial genes, and things for the plant to select. And the plant does select from this community. So we have this huge diversity, but the diversity is not passive. It's not just sitting there providing benefits to the plant, and if the plant's planted in a lucky place, then it will grow better. There is certainly an element of that. Certain soils are better for certain plants, but the plant is itself actively communicating with this environment, and it's shaping the environment to its own ends. So if you just take some soil and you plant, in this case, a model organism, I believe this was, which I can remember offhand, I think this is Arabidopsis, but don't quote me on that. What you find is planting any plant in some soil, you will see a selective enrichment of certain species. For example, here's Pseudomonas, strongly enriched, and you'll see other species which are found less frequently in the rhizosphere soil than they are in the bulk soil. So the plant is selecting some things and it's saying to other things, I don't want you. And what you see by and large, and this is a kind of rule of thumb, is that proteobacteria will be selected in the rhizosphere and actinobacteria will be selected against. And so this research came out and it showed that, yes, indeed, this huge diversity of bugs is strongly selected by the plant. And then about a year later, and around this time, there were one or two other studies, but people started to show that this was as a consequence of the secretion of chemicals from the plant into the surrounding soil. So this research was done by colleagues of mine at John Innes, the Osborne group and the BI group at CPAMS, and they were able to show that particular plant root metabolites, venosins, I believe, are secreted into the surrounding soil from the plant, and if you take these venosins, you can see differences in the overall microbial community, but then the different bugs are better or worse able to grow on media containing these root metabolites. So they were able to show that these proteobacteria, their growth is promoted by the presence of these plant-secreted chemicals, while these actinobacteria, their growth was retarded. So what we have here is really strong evidence that the microbial community is being shaped by the plant and by things that the plant's secreting into the rhizosphere. So, big question, why do this? Why, if you are a plant, would you spend 20% of your fixed carbon? Sorry, my light's just gone out. Perfect time for that to happen. The lights here are on a sensor. So why, if you are a plant, would you bother to put 20% of your fixed carbon into the environment to shape the microbial community associated with you? Well, there are a couple of big reasons why you would do this. The first of which is an array of different things, but I'm lumping them together as biofertilization. So biofertilization is taking things that the plant can't easily get itself, for example, organic phosphate or fixed nitrogen or certain micronutrients, and it's getting microbes within the soil, bacteria and fungi in the main, to present them for the plant. One of the big examples being arbuscular mycorrhizal fungi. So arbuscular mycorrhizal fungi are these fungi that are associated with the roots of plants. And as came up in the quiz earlier, they're found in interactions with about 90% of all plant species on Earth. They're incredibly important interactions. And they're called arbuscular mycorrhizal fungi because the interaction surfaces within plant cells look like these little trees or arbustles. And so that's the reason for the name. But these fungi engage in a bidirectional interaction with the plant, where they get carbon, fixed carbon from the plant, and in return they provide the plant with micronutrients and with phosphates. So this is a really important example of biofertilization. You will find similar interactions in the case of nitrogen-fixing rhizobia. So these are bacteria that interact with plants and fix nitrogen. So lots of legume plants, as Sharon was describing earlier, will form symbiotic relationships with soil bacteria, and they will be called things like rhizobia, cynorhizobia. These bacteria will interact with the plant. There is a signaling interaction using nod factors, and this will result in these bacteria entering the plant through these root hairs and forming these specialized compartments called nodules. And these nodules are stuffed full of bacteroids, which are an evolved form of bacteria that engages in nitrogen fixation. It takes atmospheric nitrogen, converts it to ammonia that can be used by the plant. And again, this is an incredibly important form of, it's one of the major ways that nitrogen is presented to agriculture. And prior to the Haber-Bosch process, it was the primary mechanism for getting nitrogen into soil and into agriculture. But these aren't the only mechanisms, these irreversible symbioses. You'll also find biofertilizing bacteria. So these are, yes, symbiotic and nitrogen-fixing bacteria like Rhizobium and Bradyrhizobium, Cynorhizobium. You will also find free-living nitrogen biofertilizers, for example, Azospirillum, Azotobacter, and these are bacteria that will live freely around the roots of plants, and they will engage in nitrogen fixation, and they can enrich the soil for nitrogen. They're doing it primarily for themselves, but the plant will get benefit if it's surrounded by these nitrogen-fixing bacteria. And you will also find an array of different bacteria that will solubilize inorganic phosphate, which can be presented to the plant in the form of amino acids and organic acids. So, for example, you will have Pantoea, Microbacterium, Pseudomonas species that will engage in phosphate solubilization. So this is biofertilization. You'll have lots of different species doing it, and lots of important inaccessible nutrients can be presented to the plant. Then there's another major group of reasons why plants might do this, and I would lump those together into growth promotion and stress control. So here is an example where the plant is not taking nutrients as a consequence of its interactions, but it's having its behavior manipulated by the bacteria surrounding it in ways that are positive for the plant. So an example of this is in the production and the deployment of auxins. So auxins are plant growth hormones that promote plant growth and development. If you add auxins to the base of a cut-off seedling, you'll see root development. If you add auxin to seedless strawberries, then you will get fruit development. And these auxins, generally, there are different auxins, but by and large, what most people refer to when they say auxin is indole 3-acetic acid. And this molecule, IAA, is produced by a number of different bacteria. So bacteria like Azotobacter and Pseudomonas species will encode genes for the production and degradation of IAA, and they will produce this molecule, even though they don't use it themselves. They'll produce it in order to promote plant growth. That benefits the bacteria, which get more exudates, and the plant, which grows more strongly. And they also will produce lots of other plant growth factors like gibberellins and cytokinins. Bacteria in the soil have also been shown, so species like Enterobacter and Bacillus and certain other Pseudomonas species have been shown to produce volatile molecules like 2,3-butane-diol and acetone. And these are plant growth promoting metabolites. They've also been shown to produce molecules like 2,4-DAPG and pyrroloquinoline quinone, or PQQ, which are antifungal and antioxidant molecules that will trigger and promote plant immune system responses to pathogens, but also have been linked to plant growth promotion. So there's lots of different molecules being produced by these different soil bugs in order to promote growth. We also find examples where these bacteria will control stress within plants. So if you are a plant and you undergo a stress response, you're hit by some toxic chemicals or a pathogen or heat or salt or something like that, then you'll produce a molecule called ethylene. And this ethylene will inhibit your root growth and it will trigger aging and senescence within the plant. Now, obviously, if you're a soil bacterium, you don't want this to happen. You want your exudates to keep coming. So you will produce an enzyme called ACC deaminase. An ACC deaminase is an enzyme that will short circuit this ACC synthase and ACC oxidase to ethylene pathway. So this is a biochemical pathway within the plant that leads to stress responses. And these bacteria, by producing ACC deaminase, can short circuit this pathway and make the plant think that there is no stress. And this will enable plants to keep growing strongly in the presence of hydrocarbons or drought or salt or heavy metals. This isn't necessarily advantageous for the individual plant, but in the context of agriculture, this will enable us to continue growing plants on soil that perhaps would be stressful for the plant otherwise. So how does all this fit into my research? Well, what we're interested in my lab is taking these different processes, all of these different plant growth promoting and stress tolerance systems, which are all present in some of these bugs. And we're interested in understanding, okay, well, which ones are important in which context, which ones are selected in which environments, and can we manipulate this in order to enable plant growth? So just in the last couple of minutes, I'll just give a very quick example of some recent research we did in this area where we found some quite exciting results. So this is a project looking at two different varieties of barley, Chevalier and Tipple. Chevalier being a pre-Green Revolution variety and Tipple being a post-Green Revolution variety. This work was done by Alba Pacheco Moreno in my lab in collaboration with a company called New Heritage Barley. And what we did here was grow these different barley varieties, which all look very different in the field, in the same soil. And then we looked at the bugs that had been selected by these different plants. And what we found was if you look at the abundance of different microbes within the soil, you see that there is quite a lot of shaping going on in both Chevalier and Tipple. But the rhizosphere and root-associated Pseudomonas populations are quite different in their abundance, along with another couple of species like Massilia. But we looked at the Pseudomonas in particular, and what we found was that there is distinct phenotypic differences between the bugs that are recruited by the roots of these two different barley varieties. So the Chevalier population looks quite different to the Tipple-selected Pseudomonas population on these selected plates. And when you look at their genotypes, you find that exactly the same applies. You have distinct clustering by the plant genotype. So if the plant you grew was Chevalier, then these genotypes cluster together from the recruited bugs, whereas the Tipple selected bugs cluster together. And this seems to happen time and again, that you're seeing distinct plant recruitment of particular genotypes of bugs. And these genotypes will be associated with different phenotypes and with different clusters of bacterial genes. Now, I don't have time to go into all the details of this. I can say that this work was published last year in PLOS Biology, if you're interested in reading it. What I can say is that this matters for the plant's growth and for plant growth promotion. So if you take Chevalier and Tipple and grow them for 4 weeks in the same soil, and you isolate the entire rhizosphere microbiome, and then re-inoculate Chevalier and tipple plants in sterile growth conditions with those different microbiota, what you find is that if you give Chevalier back its own microbiota, you get a substantial increase. So this is the pre-Green Revolution variety. You see a substantial increase in plant dry weight after 4 weeks of growth. You get an increase, but a less obvious one if you give it the elite microbiota. If you give tipple, the elite variety, its own microbes, you don't get much of an advantage. And if you give tipple the pre-Green Revolution varieties microbes, you get a growth defect. So it looks like there's a distinct genotype specific growth promotion advantage to be had here. So what we want to do going forward is really to understand this in more detail and to understand why these bugs are beneficial and why these plants are selecting them. And that's what most of the research that we do is now focused on. So that's all from me. Thanks for listening, and to my lab members and collaborators and funders. I would say that Rosaria, the PfBIO, who are the company which spun out of my lab, is really taking a lot of this work forward. So I won't talk too much about it now, but I'm sure you'll hear more about the sort of approach we take in the talk from Rosaria. So thanks for listening.
Thank you, Jacob. Very interesting and clear presentation. Very interesting concepts, and best of luck with your research, because it seems like it's very aligned with what we need for a more sustainable agriculture. Thank you so much. So I have a couple of questions for you. One relates straight to your results. So can you just give us an idea how environmental stressors, for example, things like drought or soil salinity, can alter, the root exudates profiles, and how this can affect the plant ability to recruit these stress mitigating microbes that you were detailing?
So I would say that plants, you will obviously have plant adaptation to different stressors. So you will have plants that are better able to live in, for example, a salty environment. By and large, if a plant is exposed to a stress that it isn't adapted to, it will respond by shutting down. So it typically responds by producing ethylene and or triggering its immune responses. So if it's a perceived threat, so if there's a pathogen, for example, then the plant will respond by triggering either a systemic immune response to that pathogen, or it will trigger its induced systemic resistance, which is something where positive bacteria, so commensal bacteria, can pre-warn the plant that there may be a pathogen out there. Mm-hmm. So plants tend to respond by going into a kind of resistance mode, which will inhibit their growth. And so it's important not to think of the bacteria as being necessarily friendly, that they're not friendly. When they're doing things like shutting off stress responses and promoting plant growth, they're very much doing it because that means they get more exudates. And so when we talk about bacteria as being plant growth promoting or stress resolving bacteria, that's something which is not, it's good for agriculture, but it's not necessarily good for individual plants.
So does it mean, sorry, just does it mean that the plant will suffer later on, that it can eventually collapse because it's not acting according to the responses from the environment?
I suppose in extreme circumstances, yes. All of these pathways are present in plants to protect the plant. So obviously, crops that we grow in the field aren't necessarily things, modern wheat is a very, very long way from a plant that would grow in the wild. And it's best not to think of the sorts of crops that we grow as being in any way natural. They're very strange things, really. So I suppose if in certain circumstances, yes, the plants would hope to last out a particular stress by senescing. And if they're forced to not senesce, then that may be detrimental to them in the long term.
Okay, that's a very interesting perspective. Thanks for that clarification. Jacob, we also have a lot of practitioners and hands-on farmers. So what practical steps do you think farmers should take to leverage on these plant micro partnerships to enhance what we discussed here in nutrient uptake and disease resistance?
I think there are two really, well, maybe there are three, I would say, really promising routes for delivery on this kind of research. The first one is the production of microbes that will contribute to, or microbial products that will contribute to plant promotion and disease suppression. And that's the work that Sharon was discussing and Ros will talk about. A second one is breeding new varieties that will recruit these bugs in the first place. And I think that is promising for certain crops. So, for example, for cereals, crops where there is a really strong breeding element in their production already, and lots of tools to identify genes that will recruit particular bacteria that we're interested in. So crops such as wheat and barley, I think there's lots of promise for breeding for beneficial associations. And then the third pillar will be advice to farmers and changing land use practices. So there are examples already, for example, the production of suppressive soils. If you grow wheat in a monoculture for several years, then you will start off with a buildup of Galliminomyces, the take-all pathogen in the soil. But then after about 3 years or 4 years, that will then become suppressed. And that's as a consequence of the buildup of pseudomonads within the soil that will suppress the pathogen. So that's the kind of, I think increased research in this area will lead to better advice to be given to farmers. It's fair to say that this whole field is still quite new in many ways. We've only really been able to understand these things at a molecular level for about 10 years. So I think we're in a very exciting period in terms of really getting a molecular level understanding of what's going on.
Yes, it's very interesting, and we definitely need new solutions because things are getting tougher. Jacob, a couple of questions from our participants. Maria has asked a question. When we're talking about poor soils, as in the case of the soils here in Portugal, which are scatter and poor, so poor soils, low organic matter, do you think that plants are capable of making a good recruitment of microbes?
Yes, absolutely. I think they are. I think all of the carbon in the environment, like in the plant microbe environment, like 100% of it comes from the plant from fixed carbon dioxide. Plants are remarkable things. So long as they have access to nitrogen and to phosphate, then they will be able to essentially just build a plant out of nothing, and they can then feed the microbes, and you will get a virtuous circle. So long as certain key nutrients are present, then plants can enrich the soil, absolutely. I think a lot of modern agriculture is extractive, and that's in part because intensively bred varieties of crops and certain certain farming processes like tilling soil lead to soil degradation, but that doesn't mean that process can't be reversed.
It's very good to end with a note of hope. Thank you, Jacob. One final question. Can we say that biofertilizer mycorrhiza, due to its adaptability to a wide range of crop, is better than bacteria, which might be crop specific? And you're smiling.
I think it's fair to say that mycorrhiza as a group interact with 90% plus of plants, but that doesn't mean that those interactions aren't also specific. You will get specific interactions between different mycorrhiza and different plants. Certainly mycorrhiza are really important. One thing that is I guess underappreciated is that if you add phosphate to a field, the mycorrhizal interaction with the plant will shut down. So the plant will only engage in AMF symbiosis if it doesn't have access to artificial phosphate fertilizer. I think there is absolute advantages to both bacteria and to fungi, depending on the context. And there's all sorts of reasons for that, like how easy they are to handle, their range. I think there's room for both, I would say.
Okay. Thank you for your advice. Thank you for sharing your expertise. Jacob, we need to move on. Time runs very fast. So I ask you to stay with us, but now I will call Rosaria Campilongo to the floor. And let me just introduce you, Rosaria. She's the co-founder, the director, and the CEO of PfBIO, which is a pioneering company in microbial-based crop protection solution. Rosaria holds a PhD in molecular microbiology from Sapienza Università di Roma and has extensive experience in both academic research and agri-tech industry. And it was in 2022, as we've just heard, that Rosaria co-founded PfBIO alongside two colleagues, spinning it out from John Innes Centre in Norwich, UK, where she had previously conducted postdoctoral research. Under her leadership, PfBIO developed Bio-Sift, an innovative platform that identifies natural biocontrol bacteria to create environmentally sustainable solutions for plant health and crop protection. Rosaria has been instrumental in securing funding through grants and private investments, building a top-tier team and driving the company's growth in the agri-tech sector. And Rosaria, it's a pleasure to have an entrepreneurial woman also here in our webinar series. Welcome, Rosario. The floor is now yours.
Thank you very much. Thank you very much for the kind introduction. I'll share my screen. Just a second. Can you see my screen now?
Yes.
Just a second. I'll move you here. Okay.
Perfect.
You should see the full screen now, right?
Yes.
Okay. Yes. Well, thank you very much. Thank you very much for organizing this and for inviting me. My talk, I think, will be a bit less scientific, but I guess it will give you an idea how to translate the research from the lab to the real world. So I'm very happy to be here and to introduce PfBIO to you and what we do. PfBIO, as has been said, is a spin-out company from the John Innes Centre, and we are based in the UK in Norwich Research Park. And what we do, we sift nature for agricultural lies to advance biological solutions and promote sustainable agriculture. Okay, so PfBIO has 3 directors and co-founders. There is me, Jake, that give a great presentation, and another colleague that is called Andrew Truman. Jake and Andy are both group leaders at the John Innes Centre. I'm the CEO, and then we have Francesca and Benny that works in the R&D, so they're advancing multiple projects. And then we have Jonathan Clark and Sky Van Heiden that are providing business guidance, and they're connecting us with valuable people working in these sectors. And we are very happy to been supported by several sponsors, including Innovate UK and Anglia Capital Group. Oops, sorry, it doesn't work. Okay, so, but why do we exist? You probably already know, but I'll say that anyway. So the use of pesticides and other agrochemicals has been raising concerns for the environment and human health. And so the European countries, in response to this, have introduced several policies aiming to reduce and halve the use of pesticides by 2030. Many of the most hazardous active ingredients have also been banned from the market. That left growers with a few options that they can use to protect their crops and optimize the yield. At the same time, there has been also the emergence of new plant disease, all driven by climate change. And so we want to support farmers and sustainable agriculture during this legislation shift, and by providing environmentally friendly and sustainable microbial solution to optimize crop production as an alternative to synthetic chemicals. And we want to bio-sustain one crop at a time. This is our mission. With a vision to become a leader in these sectors, develop a biosolution that can reshape farming, reduce chemical reliance, and so improve food security to build an healthier planet. So with climate change and push for net zero, and as I say, with this shift in legislation in many European countries, and then a high demand for organic food and more sustainable way of farming, the market of biocontrols and biostimulants is expanding rapidly. However, there is still a significant gap of effective solutions that can meet farmers' needs. And this gap, in combination with a fragmented market, represents a great opportunity for PfBIO. The biocontrol and biostimulants market together, they have a value of $12.5 billion. If we focus only on the microbial segment, these have a value approximately of $4 billion. We want to capture a market of $50 million in 10 years' time, but just licensing some of our IP and selling our first range of biostimulants and bioprotectants. But how we do this? We developed a discovery pipeline called BioSift. BioSift allows us to quickly identify the most suitable bacterial strains that we can use to create our product and to unlock the mode of action. Everything starting from farmland and environment. So we do sampling the environment, and then in the lab we do selection screening. We unlock the mechanism of action of these strains using specific tools that includes also bioinformatics and chemistry, and then we validate the performance of the promising candidate in plants. We start with small-scale experiment in-house, and then, of course, we will take everything in the real world, working with partners that can run field trials for us. So BioSift is 5 times faster than conventional discovery platforms, so allow us to quickly identify the most suitable strains. It's also cost-effective and is so very efficient. And while the process remains a trade secret of the company, the output of the process are patentable solution. And this includes microbial strains, the extract, and some genetic information. So at the moment, we are very focused in developing bioproducts tailored to the berry sectors. So we are creating MAR-A and MAR-B that are multi-active, robust antimicrobials and biostimulants. So our solution utilizes patented microbial strains with properties that target pathogenic fungi, oomycete, and bacteria, and they can also provide biostimulants benefits. So far, we raised over $350K to work on this project from grants and private investment. Now, last summer, we worked with Agri and IAB here in the UK, so we performed our first field trials. And to focus on the biostimulant side of the project, we tested several strains to check if they could have an effect on the boost of strawberry yield. And we see that one of the strains were able to boost yield by 18%. So to better understand this market and the needs, of course, I'm always engaged with berry growers. So far, I create connection here in the UK, but I would be very happy to create connection across Europe, in Portugal as well. So I'm very open to reach out and be connected with our berry growers, because I really would like to test our solution and validate it. So another strain that we identified in-house has instead the ability to promote root development. Originally, we thought that this strain, this effect would have translated in a better yield after field trials, but then when we test both strains, we realized that actually the yield boost of these strains was not really translating in a better yield, while the other strain that also had an effect on root development, but in a less extent, was actually boosting yield. So in the end, we decided to actually create a product for nursery and a product for production side. The product for nursery, that we will call here MAR rooting, show a 17.7% root increase from when we apply the strain to strawberry runners, okay, in comparison to untreated strawberry runners. While, as I said before, the other strain has the potential to boost yield by 18%. However, both strains have the ability to make the plants resilient to crown rot by respectively by 80 and 90%. And so this is a great advantage for both stage during strawberry plants growth, because of course you want to generate strong plants at nursery stage. They can also have not any where you can limit it the concentration of spores or the concentration of Phytophthora cactorum. And so you can generate high quality plants that then you can sell to growers that in this case we move to the production site, that they can actually use all the plants that they buy to generate crops. Because what's happened is when with crown rot you can lose between 10-30% of plants during production. So yeah, we are working on this the development of this two biosolution, and of course we are looking also at some technicality of the product. It's fair to say that we apply the strains as a soil drench. So what we checked the survivability of these strains at the root side, so in the rhizosphere. And we applied every month as we realized that the concentration of the strain was didn't decline so much in this time, and so the strain was actually able to support and promote plant health during this time. So at the same time, also we are looking at the shelf life of the product and the stability of the strains, because you can have the best strains that can stimulate the plants or protect the plants from pathogens. But if you're not able to actually translate that in a product that has a good shelf life, well, you cannot commercialize it. So we have done some initial study in-house where we create a liquid and powder formulation. And although here is still missing the last point that we just generated a couple of days ago, we follow for like 6 months. And in the end, we realized that in our case, the powder formulation would be the best formulation, so the best way to make our product and sell it. And it seems like it's more stable when a low temperature compared to room temperature. In that case, otherwise, you will start to have a decline of the population. However, what we are doing now, we are working with specialist partners, specialists in formulation, to improve the stabilization of the strains and the shelf life, so that the product will be stable at room temperature for a much longer time. We are aiming for 18-24 months. At the same time, we are then going to validate the final formulation by performing field trials here in the UK and hopefully soon across Europe as well. After the validation phase, we will start to seek regulatory approval in strategic European countries. we would probably go for national regulation approval. So we are targeting country where there is a quite big good production of strawberries, including Spain and Poland, that are one of the first producers and exporters. But then this also will include France, Germany, Italy, Portugal, and the Netherlands, of course. So, but we are also working on other projects. With BioSift, we are identifying new bioactive microbial strains and their mode of action. And so we are advancing multiple projects, as I said before, all on our mission to biosustain one crop at a time. These are other crops of interest for us. And we are working on developing biosolution for onions, hops, and lavender in collaboration with VCS Agronomist Group that is based here in Norfolk. Just today, we're actually starting our field trials in onion. But just to give you like a brief taste on the biocontrol side of our project, we are working on potatoes. So we identified several strains that have activities against some pathogens that are relevant in this field. They can, in this case, for example, DK solani, Rhizoctonia solani, they can cause rot. And we did some plant tests, and in this example, we see that strain number 3 has great potential in protecting the potatoes against the D. solani stress. And we have 60% less rot in tubers when the potatoes were treated with these strains, and almost no symptoms at the stem of the plants. And then we also look at the Rhizoctonia solani, and among all the other strains we test, although the strain number 3 also in this case has a good effect in protecting the potatoes against the stress, we also see that strain number 40 was actually performed very well. And in this case, we will take this strain forward to create a product to protect potatoes from rot. And another example I want to give you is about the onions. Fusarium is a big problem in onion cultivation. So we isolate Fusarium oxysporum strains from infected onions from a field here in Norfolk. And then we screen our collection to identify strains that have activities against this pathogen. We found several, so here there are some representative where you could see some have activities, some others, they don't show any activities instead. And we did some tests in-house on onions plants, and we identified this strain C4 that actually increased the resilience of Fusarium basal rot by 84%. And now we just started field trials today with VCS and test the same strain in field trials in a commercial environment. And yeah, I just want to conclude saying that we are very open to collaborate with other farmers and working on different problems that are actually real commercial problems in the real world, and translated our lab research in something that can be very useful to support sustainable farming. So I want to thank you very much.
Thank you, Rosaria.
Yeah, if you have any questions, just let me know.
Thank you so much. It's been great to hear that good things are coming from the lab. Also, to offer our test beds here in Portugal, we do have a program that is just aimed at testing new technologies in fields, and also to remind you that Portugal is a big strawberry producer. We are suppliers of Wimbledon, so you know,
Okay.
if not in quantity, at least in quality. We are very well known about our strawberries production. So if you want to approach, if it is one of the options, we're sure open to discuss, and I'm very glad to test your products in the field.
That would be great.
Thing
Thank
about de nada. So you mentioned, just a quick question, you mentioned yield boost of almost 18%. Can you just detail the conditions of the trial that led to this increase?
Of course, the strawberry can be grown in a very different way, I would say, across Europe, and maybe in the US is also different. That's what I know. In the UK, that is how we perform the trials. They are grown in a soilless media that is called coir, this coconut pellet, and they use basically coir bags. so that we can control the quantity of water and all the nutrients that they put in. And they are usually grow under polytunnels, unless they are grows in glass houses to extend the production season. So these are the conditions in which it was as a soil drench application every month for 3 applications in total. And well, we did it in summertime, of course. We did it last summer between July and September. Summer last year in the UK was not great, so I didn't have much expectation actually. And but I know, for example, in my country, in Italy, many strawberries are grown directly on the soil. So that will be slightly different. So that's why I'd like to also check if our solution works as well in a different kind of environment, a commercial environment. Although it's true that many growers kind of start to use soilless media as well, or bags, because that would allow them to use less water. So to reduce the quantity of water, especially during summertime, where there are regions where, you know, it's quite, there is not so much water available in a way. So
yes, thank you, Rosaria. We resonate well with both scenarios. We also have the two modes of production here in Portugal, and the question of water for us it's critical. Although this winter doesn't stop raining, but
yeah, I think. But then in summertime is always going to be critical anyway.
Yes, exactly. Okay, very conscious of our time. I will just have one final question for the 3 of you, because fortunately our time runs very fast, and this is from a practical point of view for our farmers. I know that you have done more detailed research, but can you just share with us your perspective on the potential synergistic effects between the different types of biostimulants? So using microbial inoculants, seedweed extracts, humic substances. Can you detail how all this combination works in terms of if they are applied to seeds or to the plants?
Just share my view. I think that they cannot all be winners in a way, but you will never know until you actually do a test to see if there is like synergy between among the products, because in terms of like for the biologicals at least, they can be very well, they're kind of individuals, right? So they behave in a different way, so they may be happy or not when you also provide to the plants completely different nutrients or seed extract, plant extract. So it needs to be tested. And I know agronomy-based companies do that. They actually love to see if there is synergy between products and so on. So yeah, that's what I think, that there can be a victory there in using them together, but until you don't try, you don't actually design a specific plan, it may be difficult to actually answer this question. Yeah.
Okay. Thank you, Rosaria. Jacob, I see you nodding. So
I mean, yeah, I would absolutely agree with that. I think the answer to your question is it depends. There's so much complexity in these interactions. By and large, if you are providing additional nutrients to a plant, that might be beneficial, but it may shut off some symbioses that would provide them from less nutrient-rich soils at the same time. So all of these different, certainly all of the living organisms, so the microbes involved in biostimulation, need to be understood as living things, and they're complex and they will have lots of different inputs. I think at this stage, we don't really have the understanding of how to predictably combine them, but that's not to say that you can't make combinations in the future that would be useful. I think it just requires research.
Yes, and then we have AI to assist us, and hopefully we don't need to make decisions. The machine will do it for us.
Maybe.
Maybe. Sharon, what is your view?
Yes, I agree that if you're adding a nutrient-rich seaweed extract at the same time that you're adding the bacteria, then you may prevent that symbiosis from even developing. So maybe add the extract later if it's needed. But I think, give the microbes a chance first. And having a mix of microbes, I think, is a good solution.
Okay, good. Thank you. So, Sharon, you have a question in the chat. If you can just spare a couple of minutes in the chat, I need to move on. Thank you so much. But before you all go, we have prepared a short questionnaire for you. You know this. If you want to have our certificate, please use this QR code. You will have a very short quiz based on the presentations that we have here today. I think this was indeed a great webinar. I think we've learned a lot. We see very serious people doing some very serious work. We will definitely be on the lookout what's coming from your end. So thank you so much for being here and share your research and your work with us. For those of you that want to use the link is here, but let me just to remind you, this is not the last session. We still have one other session coming next week. So we will look at innovations in soil. We will continue to talk about biostimulants, trying to understand what they really are, how they work for farmers. So I hope that you can all join us next Thursday, same time, same place. So thank you so much. It was really a pleasure to conduct this webinar for you. Thank you all.